Hi, everybody, and thanks for the nice introduction. I'm very happy to be here today to present one of my latest published papers. It was published in January 2024 on gut microbes. I would like actually to start from the end of my talk by thanking the organizing committee for this great opportunity of being here today to present, and all my co-authors that you can see in these slides. This was a great collaboration we had that led to the publication of this study. I am pretty sure you have heard already about the word anorexia nervosa, but I would like to start my presentation by briefly introducing this condition. It's the third most common chronic disease in adolescents and the deadliest of all psychiatric diseases. It's characterized by a very disrupted feeding behavior. So patients diagnosed with anorexia nervosa have an insufficient energy intake, very low amount of food they eat, and this is causing then a very low body weight. Patients also have a distorted body image and fear of gaining weight. The treatments usually includes weight restoration and psychotherapy. This can be done at the hospital. This is called inpatient treatment, or while the patient is at home. This is so called outpatient treatment. The big issue about this condition is that the treatment is often insufficient and there is a very high relapse rate. Despite being very common as a condition, the underlying pathophysiology of the disease is poorly understood. So the aim of my study is to identify in the gut of these patients microbial markers that could help in perform an early diagnosis of the disease, inform the prognosis of anorexia, and also, why not, identify some targets that could be kind of beneficial bacteria that can be used as a supporting therapy for anorexia nervosa. And to try to reach these aims, I benefited of a great collaborations with clinicians and psychiatrists at Uniclinic in Aachen. In the map here, you can see where Aachen is in Germany. In Aachen, our collaborators work in anorexia nervosa ward that is accepting adolescents that are diagnosed with anorexia nervosa. The patients usually are between 12 and years old, and they access the clinic and they stay until they reach a BMI that is considered healthy. Thanks to this collaboration, we had the possibility to collect fecal samples from 56 female patients and 34 healthy controls that were age and sex matched. As it is an inpatient treatment procedure, we were able also to collect samples longitudinally up to 9 time points until discharge. That's when the patients reach a healthy BMI and they can go home. The advantage of our study is that we also have access to an extra time point, and this is this is the first time in literature that was reported. In fact, the patients and the healthy controls visited the clinic 1 year after admission. Here, the patients were divided into groups. Low weight patients, that are the ones who have still a BMI below the threshold, and weight recovered patients that still have a healthy BMI. For all these samples, we performed total DNA extraction and we analyzed microbial communities by using 16S amplicon sequencing. We first looked at how alpha diversity changed over time. As you can see in these two graphs, alpha diversity is quite stable during hospitalizations, both when looking at Shannon index and Chow index. However, we see the lowest alpha diversity after discharge at 1 year follow-up. I have to admit that actually this, the alpha diversity of patients that are weight recovered is not significantly different from the one of healthy control, but the p-value is actually very close to 0.05. We then used beta diversity measures to understand whether the microbiome of patients is actually normalizing during weight rehabilitation. And that means, is it becoming more similar to the healthy controls? So in this plot, you can find a partially square discrimination analysis reporting the microbiome of patients and healthy controls at admission, at baseline. And we found a significant difference at lower taxonomic levels. But what happens after weight gain rehabilitation? We find that there are still some differences in the microbiome. However, these are much smaller than they were before. We then tested what happens at 1 year after this long-term weight gain. We had to divide, of course, the patients in low weight and recovered. And we found that when taking into account the three groups, we find residual differences in the microbiome at all taxonomic level. However, when we split the analysis in two groups by dividing low weight and weight recovered, we found that actually the residual differences for the weight recovered patients are very small and only at ASV level. We also exploited the longitudinal nature of our study to understand how microbiome changes during hospitalization and weight recovery. In this plot, you can find a multi-level longitudinal partial list square discrimination analysis reporting all the fecal samples of all the patients at all the time points. At the beginning, so the admission samples are in blue, dark blue, and the 1 year follow-up samples are in dark red. And you can actually appreciate a clear shift in the microbiome composition from admission to 1 year. This is actually supported also statistically. We performed a Permanova on the Bray-Curtis dissimilarity index by comparing patients at admissions, discharge, and 1 year follow-up, and we found a significant p-value suggesting that this shift is actually there. This was true not only at ASV level, but also at all taxonomic levels. We performed also pairwise comparisons at all time points, but I don't have time to explain this to you now. Please check our paper if you're curious about this. And finally, we also wanted to understand whether some members of the microbiome at admission could be relevant for the clinical outcome. So the main question is, can some members of the microbiome present at admission predict longer inpatient treatment, so more difficult treatment in the hospital, or a higher BMI at 1 year follow-up, so a better recovery? We found a limited number of taxa that were actually predicting clinical outcome. One of these is Sutterella. The relative abundances of Sutterella at admission was significantly correlated, positively correlated to the BMI at 1 year. And Sutterella is known to be positive commensal with anti-inflammatory properties in other conditions. So we would like to confirm this positive role in another study and hopefully use this as a potential probiotic. So to conclude, we observed reduced but ongoing alterations in the gut microbiome after 1 year follow-up, also in the weight recovered patients. And we reported that the microbiome at admission has a prognostic value for the course and outcome of the disease. This is actually very interesting because we could then tailor the therapy during the inpatient treatment based on the needs of the single patients. Of course, we also hope that we could confirm the importance of Sutterella so that we could use this as a supporting therapy for anorexia nervosa. We actually have some strengths and limitations in our study. The sample size is very small. We are currently working with a bigger data set with patients and heavy controls coming from 3 European cities. And of course, the big limitation is the use of the methods. That was actually interesting for us because it allowed us to sequence a very high number of samples. So from the economical point of view, it was the only choice we had. But of course, using shotgun metagenomic sequencing could have helped in having a better resolution in our results. And with this, I would like to thank you for your attention, our funders, and to all my collaborators, but most importantly, all the participants of the studies and their families. And I will be happy to discuss more at the Q&A session after the other selected talks.